Let's assume this is a cell, and this portion of the cell is B cell, and this portion of the cell is macrophage or dendritic cells. In case of B cell, two antibody actually bind with the plasma membrane are called IgM, and another antibody that bind with the plasma membrane called IgD. These two antibody togetherly called B cell receptor. Okay, now this antibody can bind with microbes or antigen. Let's assume this antibody bind with antigen. So when antibody bind with antigen, a process occurs called receptor-mediated, as because this IgM and IgD is B cell receptor, so a process occurs called receptor-mediated endocytosis. So endocytosis occur, and what happens? This antibody along with the antigen actually enter into a vesicle. This vesicle called endocytic vesicle, and what happens? A protein actually coat these vesicles called clathrin protein. It is always occur during endocytosis. So this protein is called clathrin. Clathrin protein actually coat the endocytic vesicles. Now in these cells, in B cells and macrophage and dendritic cells, both these cells contain another vesicles already present, that is called early endosome. This early endosome contain some enzyme. These enzymes are inactive in form because in the early endosome the pH of the early endosome is 6 to 6.5. In that pH condition, this enzyme are called protease enzyme. This protease enzyme, example is cysteine protease. Cysteine protease means the enzyme catalytic site of the enzyme contains cysteine amino acid. So this cysteine protease is inactive in form. So they are inactive. Why they are inactive? Because this type of enzyme can be activated in acidic pH, means lower pH. When the pH becomes 4 or 4.5, this enzyme becomes very active. Now it is inactive in form. This is called early endosome. This vesicles is already present in the B cell, macrophage and dendritic cells. So what happens when endocytic vesicles enter into the cell? The clathrin protein become uncoated. Now the endocytic vesicles is uncoated by the clathrin, so there is no clathrin protein. Now in this situation, the endocytic vesicles can fuse with the early endosome. When these endocytic vesicles fuse with the early endosome, now the antibody and the antigen is present in the early endosome. Okay, it is fused. So this process is called endocytosis. Okay, so this is the first condition or first way. In case of macrophage and dendritic cell, one thing occurs that is called phagocytosis. What happens? These macrophage dendritic cells form a pseudopod-like structure in which pathogen or like bacteria are engulfed. So when these pseudopod actually merge with each other, they form a vesicle-like structure. This is called phagosome, and inside the phagosome bacteria is present. It can be bacteria, protozoa, whatever may be. This process is called phagocytosis. So this is the second way a antigen or pathogen can enter into the cell. This is called exogenous pathway because in this case MHC2 molecule actually present this type of antigen or peptide. So this is phagocytosis. Another thing can occur. This is called autophagosome. What happens? A cell can autophagy its cell organelles or its protein. So in the autophagosome, cell proteins, these are the cell proteins are present, or organelles proteins. So these are the cell protein, autophagy. So this is the third process. So this endocytosis, receptor-mediated endocytosis, phagocytosis and the autophagy, by all this way actually vesicles are formed, like endocytic vesicles. Phagosome is also a vesicle and autophagosome is a vesicle. All these phagosome and autophagosome also fuse with the early endosome. So they all fuse with the early endosome. So what happens? This bacteria now will present in this early endosome. These cell proteins can also be present in this early endosome. As the early endosome pH is somehow neutral or high, then this protease is actually inactive in form. But in this early endosome, a protein is present called, this protein is called V-ATPase. This protein or molecule actually lowers the pH of the early endosome. Now, when this protein actually lowers the early endosome, the early endosome becomes late endosome. By this V-ATPase, the pH becomes 4.5 to 5. Now these vesicles transform into late endosome, and all the protease that was inactive in form becomes active. So they can cleave the antigen or bacteria and make this antigen or bacteria into fragment. So they make it fragment. Okay, so these are the exogenous peptide. These are the exogenous peptide, and these are the now active protease that can cleave the antigen or pathogen that is already present in the early endosome. One another important point is, when early endosome becomes the late endosome, lysosomes, these are the lysosomes, also fuse, and lysosome also contains some proteases, many proteases. Lysosome also fuse with the late endosome. Lysosome also fuse with the late endosome. Now this lysosome containing enzyme is also present. They also cleave this product. So this is occurring, this is actually antigen processing. In case of class 2 MHC molecule, antigen are processed in these vesicles. Okay, now MHC II molecule is actually produced in the rough endoplasmic reticulum. Let's assume this is a rough endoplasmic reticulum. Okay, this is RER. So let me draw a class 2 MHC molecule. So let's assume this is a class 2 MHC molecule, this is the alpha chain and this is a beta chain, and this is a class 2 MHC molecule. Okay, but here is the problem, because in the rough endoplasmic reticulum, class 1 MHC molecule is present. This is the class 1 MHC molecule. And what happens in case of class 1 MHC molecule? The antigen or peptide loading occur in the rough endoplasmic reticulum, RER. The peptide loading, this peptide like endogenous peptide, these are the endogenous peptide coming from the cytosol. Let us assume this is a cytosol. In this cytosol, proteasomes are present. These proteasomes are cleaved the endogenous proteins into peptide fragment, and there is a gateway called trap protein. This trap protein actually helps to enter this peptide molecule into the RER. Now this peptide molecule binds with this MHC1 molecule. So antigen loading in case of MHC molecule, MHC1 molecule occur in the rough endoplasmic reticulum. So if this MHC2 molecule antigen or peptide cleft is open, if the peptide cleft is open in case of class 2 MHC molecule, what happens? This endogenous peptide can be loaded into this empty peptide cleft. So if the peptide cleft is open in case of MHC2 molecule, there is a chance that the endogenous peptide can be loaded on this MHC2 molecule. Then the function or the property of the MHC2 molecules will be destroyed, because the function of the MHC2 molecule is to present this exogenous pathway into the plasma membrane, not the endogenous peptide. So for this reason, there is a protein that actually seal the peptide binding cleft or groove. This protein is called invariant chain. This is also written as capital I, small I. It is also called CD74. You don't have to remember, I am just mentioning. So this protein actually seal the peptide cleft of the MHC2 molecule, so that no endogenous peptide can bind. Now this complex, this MHC2 complex with invariant chain come out from the RER, rough endoplasmic reticulum, to a Golgi. Why? Because all membrane bound protein actually produced in the rough endoplasmic reticulum, and it actually entered the Golgi. Okay, so the MHC2 molecule with the invariant chain now entered into the Golgi, and from the Golgi, this is the lysosome. From the Golgi, this MHC2 molecule with the invariant chain enter into the late endosome. So now you can understand the late endosome is very important. Now the MHC2 molecule is present in the late endosome. This is the class II MHC molecule, MHC2. But here also this invariant chain is present. This is the invariant chain. Very recently scientists have discovered there is another function of this invariant chain. They have found those cells do not express invariant chain. The class II MHC molecule cannot come out from the RER. They always remain in the RER. But if these cells is transfected with this invariant chain DNA, then the cell can produce invariant chain. And what happens, scientists have seen that now the MHC2 molecules is present in the late endosome. So what actually performing by this invariant chain? Invariant chain is actually guide of the MHC2 molecule. If there is no invariant chain, the MHC2 can be present in the rough endoplasmic reticulum but cannot come out to the Golgi, and from the Golgi to the late endosome. Okay, so invariant chain have a signal sequence that actually guide the MHC2 molecule from RER to Golgi, from the Golgi to the late endosome. Now what happens? In the late endosome, this invariant chain is actually cleaved. This chain is cleaved. Only this fragment, this small fragment attached with the class II MHC molecule. This fragment is not removed by this enzyme. The enzyme actually digests the invariant chain, but this portion of the fragment of the invariant chain always actually attached with this MHC2 molecule. This cleaved invariant chain is now called, this portion of the protein is called CLIP, cleave. So now this late endosome have to remove this cleave to actually attach this exogenous peptide, because this cleave have to be removed. For this program or function, another molecule is present in the late endosome that is called non-classical MHC2 molecule. So this is the cleave. So there is also a type of class II MHC molecules already present in the late endosome called HLA-DM. This is HLA-DM is a non-classical class II MHC molecule. This HLA-DM, when is very close to the MHC2 molecule, what happens? This HLA-DM helps to remove this cleave protein, exogenous protein peptide can bind in this cleft region. So HLA-DM has a positive role to remove the cleave and to load the antigen. So now you can see, in case of MHC1 molecule, the peptide loading occur in the rough endoplasmic reticulum, but in case of class II MHC molecule, the peptide loading occur in the late endosome. When the MHC molecule and the peptide is actually loaded, now this complex, this MHC2 with the peptide complex come out from the late endosome. So this complex now come out from the late endosome to the plasma membrane. So now this MHC molecule is present into the plasma membrane and is presenting the fragment or the peptide